to another Thursday monthly MSI seminar, which we're taking virtual this year. And in just a minute, I'm really pleased to welcome Dr. Shawn Campagna. And we'll have an introduction from the member of the MSI community who nominated this talk today. But first, I'll just give you a little bit of a reminder of how our talk format works, which is that we'll have this introduction and then we'll hear from Shawn for 30 to 40 minutes. And then we'll have time at the end for Q&A from everyone who is watching and participating. So feel free, if you are watching this on YouTube Live, to enter a question or comment at any time throughout the talk. And then during that Q&A portion, we'll make sure that those are, as many of those as possible at least, are communicated and discussed during that period. But without any further ado, I'll turn it over to Chelsea to introduce today's talk.
Thank you. So I'm happy to introduce Shawn Campagna as our speaker today, particularly because he sat on my dissertation committee and has continued to be a key aspect of helping me with my scientific career in many ways. So Shawn obtained his Bachelor's of Science in Chemistry from North Carolina State University in 2000. And then he completed his PhD from Princeton University in 2006, where he worked with Martin Semmelhack, Bonnie Bassler, and Frederick Hewson. He then did his postdoctoral research with Joshua Rabinowitz at the Louis Siegler Institute for Integrative Genomics at Princeton. There, he became interested in developing methods of mass spectrometric identification of molecules from bacteria and other natural resources. And this is work that his lab has continued and is now expanding upon at the University of Tennessee, Knoxville, where he has been since 2007. And he's now serving as the Associate Department Head of the Chemistry Department. Shawn, thank you so much for joining us, and you can take it away.
Yeah, thank you for the invitation, Chelsea, and everyone. It's an honor to be here. Is my screen showing okay?
Yes, I can see it.
Sounds good. Well, so thanks again. Today I'm going to talk to you about our efforts to use metabolomics to understand the function of several environmentally relevant microbial consortia. I am going to apologize in advance. In the abstract, I promised that I would talk about some of our forensics work. It turns out that since this is being recorded, I can't show you the images because there's some confidentiality reasons there. So we're just going to focus on our efforts in bioremediation today, but I hope everyone's not too disappointed because I'm excited to share this with you. So first off, let me introduce my lab. I am a chemical biologist here at the University of Tennessee, and we primarily do two things. We synthesize molecules. I started my career as an organic chemist, and then we do bioanalytical mass spectrometry to try to understand metabolism. And really the overall goals of our labs are to understand how small molecules pattern the world around us. And we do that in a variety of ways. Sometimes the molecule leads our investigation, and sometimes metabolism tells us what molecule to go after. That's the platform and the things that we look into. And really where a lot of our efforts have turned to in the last decade or so are trying to figure out better ways to use metabolomics, which is the systematic study of all of the small molecules in an organism, to really go in and figure out how the physiology of more and more complex systems are working. Now, when I say metabolomics, I'm also including the lipidome in that. I know that several disciplines split out those words, but just for simplicity, I'm really talking about any molecule with molecular weight 2000 or under when we're going to go into a lot of these investigations. And when we chat about specific vignettes, I'll tell you whether we're talking about the water-soluble component or the lipid-soluble component. Now, one of the big reasons that I got interested in metabolomics as a postdoc was because I really wanted a platform that allowed us to find new molecular structures and also to look at physiology after post-translational modification had really changed the function of the cell. And metabolomics was an avenue to do that. And so this technique really fits well into sort of the systems biology cascade, as many of you know, because it is the one technique that actually looks at the true physiological state of the system, not the information content or the cellular machinery. One of the other aspects that's exciting about metabolomics for a chemist is that the metabolome is incredibly complex. There's a lot of different molecular structure types there. And so the separation, identification, and measurement of those is actually a huge analytical feat. I'm not going to talk about that technology too in depth today, other than to say that this leads to there being a variety of methods, both untargeted and targeted methods that either look for all metabolites in the system or a subset of metabolites of interest. And that these analytical challenges are what a lot of us in my lab work on a daily basis. One of the other exciting things for us about metabolomics is that you can gain information that can answer questions at a lot of different scales. You can answer questions at the systems level where you look at the entire metabolome and try to understand how the metabolome in general is changing and whether or not you can speciate things based on the metabolome. You can look at metabolic pathway function and do in vivo biochemistry, or you can even look at single enzyme kinetics if you wanted to really go down to a fine level. And in fact, today's talk, we're going to start at the single enzyme level in the first project and then build up and show how these tools can be used to look at entire systems. It does take a lot of technology to do these experiments. And one of the things that I like to highlight that in 2007, when I began my postdoc, we were using an HPLC triple quad where we were measuring 120 metabolites of known compound or known structure. And this was taking us about 2 hours to do so. We were super excited about that at the time because the average metabolomics experiment was only measuring about 40 or so compounds in the mid 2000s. And then a really rapid expansion in mass spectrometry has led to the ability to not only identify known compounds where you have to build large libraries, but to also look at known compounds and to identify what we're going to call spectral features from unknown compounds in a single run. So currently, when we're running an experiment, instead of getting information on 120 compounds, we're now getting information on thousands to tens of thousands of compounds in every analysis. And because of upgrades in chromatographic separations, we're actually doing this in a fraction of the time that we were before. And so all of this technology has really opened up a lot of really new avenues for us, which is exciting. And going back to my beginnings as an organic chemist, I actually like the fact that we can't identify everything because it gives my students and me an opportunity to go in and find novel chemical structures. And in fact, the first project that we're going to talk about is going to be one that focuses on a metabolomics investigation that led to us actually looking into and finding a novel chemical structure. Most of the work that we're going to talk about today is in collaboration with the Loeffler Lab here in the Center for Environmental Biotechnology. And it's going to focus on bacteria that can dehalogenate compounds. The first three vignettes that we're going to go through are going to be looking at dechlorinating bacteria, and then after that, we're going to talk about one defluorinating bacterium. So this study was looking at Desulfobacter hafniase, which is a gram-positive organohalide respiring bacterium that is used in bioremediation efforts, where it's deployed in the field to clean up contamination of chlorinated compounds. Most of these pathways are cobamide, which are vitamin B12 analogs, dependent pathways. And the Loeffler lab was doing a profile of the cobamides in Desulfobacter hafnium and found that it doesn't fit into any of the known cobamides that are out there. And so here's the structure of B12, and the cobamides vary based on the structure of the lower base. To date, all of these structures in the upper right-hand corners are the ones that have been found on the lower base, and the one in the dehafniase, Desulfobacterium hafniase, seemed to be novel. So the data showing that was just initially HPLC data and then HPLC MS data, where as you can see, the hafniase cobamide doesn't coalute with any known cobamide standard. So from there, we isolated this molecule and started asking the question, what could it be? We first turned to mass spectrometry and figured out that the mass of the molecule was 1,329 Daltons and compared to a mass of 1,355 Daltons for vitamin B12. Since we assumed that the lower base would be the only thing that would be changed, as most of the cobamides use the same core and sort of linker portion, we started to assume that only the portion of the molecule here would be different. Oh, sorry, I clicked too many times. And so if we could go through and determine what the mass of this lower base was, that would give us some clue as to what the structure could be. So if you look at the dimethylbenzimidazole portion that you find on vitamin B12 and do a subtraction from that, you can easily calculate what the mass of the coronoid core itself is, which leads to just some simple math that if you take the mass of the new novel cobamide, you subtract that, or you subtract the mass of the conserved portion, you can see that the lower base should have a mass of 120 Daltons. That allowed us to go in and assign a preliminary chemical formula, which we determined to be C5H4N4. There actually aren't a lot of biological entities that are known to have this chemical formula, which led us to initially believe that this could possibly be purine. Now, this was intriguing because purine isn't found in many natural products in its unsubstituted form. And so we actually needed to go in, even though it looks simple, and do even further characterization to prove that this was the right lower base structure. Mass spec is inherently not a structural tool, but it can give us some insights on structure. And so from here, we decided to perform a labeling experiment where if you add nitrogen 15 to the bacteria as they grow, they should incorporate that into their structure selectively over nitrogen 14. And that should give a mass shift where you can calculate how many nitrogens are in your structure. If you go back to vitamin B12, you would expect 11 of the nitrogens to be conserved among all of these different cobamides, and those are the ones shown here in red, which would leave, in the case of B12, 2 nitrogens in blue that would be variable. And so if we take our new cobamide, feed it nitrogen 15 and measure the mass, you can see that there's a mass shift of 15. If you subtract the 11 nitrogens that we believe to be conserved, then these data are consistent with the lower base containing 4 nitrogens, just as purine does. So this gave us further evidence that our structural hypothesis was correct, but still we wanted to go in and get a really fine-tuned understanding of this and really nail down what this is. So from there, we did a variety of NMR experiments, which I'm not going to show you in detail here, where we took the NMR, the proton and carbon, and some 2-dimensional NMR spectra for both vitamin B12 shown in blue, and this new cobamide that we were interested in. As you can see, most of the portions of the molecules are conserved, as shown by the peaks being the same. And really the differences only come in the aromatic region from the lower bases and from a few alterations shown here in the protons in the coronoid ring. If you look at the this bacterium's cobamide, you can see that there are 3 singlets, which also corresponds with our hypothesis that there are 3 carbons that would have a proton on them. And like I said, I'm not going to go through all the details, but we were able to assign each of these signals to the isolated methylene units in the purine base. So from there, we were fairly certain that we had discovered a novel cobamide that contained purine. And the question was it biologically active? And you can overproduce the cobamides by feeding purine to a lot of these organisms. That's exactly what we did, built up a stock of the pure cobamide, and then added it to closely related species and unclosely related species to see if it had differential function between the two. The first that we added it to was this Halobacter restrictus, whose reductive dehalogenase enzyme is a 98% match to the one in the organism under study. And as you can see, vitamin B12 functions in both systems. And in this bacterium, Purine cobamide was also able to support dechlorination of perchloroethene, tetrachloroethene, and cis-DCE. If, however, we went to Helicocoides mccartyi, whose RDase match was only 20%, you can see that Purine cobamide didn't function nearly as well as vitamin B12 or other cobamides that were known to work in this system. This just highlights that the range of naturally occurring cobamides have lower base structures and lower ligand structures that are modulators of coronoid-dependent enzyme function, and different organisms need different B12 or different cobamide analogs to function in the environment. This is also one of the first few times that a non-substituted purine has been found in compounds. And so we were fairly excited about this because as bioremediation efforts continue and move forward, one of the problems is that they often stall. And one of the ways to get around that is to add a cobamide. And so understanding the suite of molecules that are needed to support dechlorination in the environment is important for making sure that you select the right additives to move forward in bioremediation efforts. As we continued our study of dechlorinating organisms with the Loeffler lab, the next problem we decided to tackle was actually looking at organisms that could degrade dichloromethane in the environment. Dichloromethane is actually still a major contaminant in the US. 260 million pounds of this are made in the US yearly. It's a major chemical that's used in a whole lot of things, from a degreaser to paint stripper. And even if you drink decaffeinated coffee and tea, the way that the caffeine is removed is by extraction with dichloromethane. And so a lot of this enters our environment every year. And one of the problems with dichloromethane is, although many bacteria are known to do aerobic degradation of the molecule, because it is more dense than water, when contamination occurs, you tend to have the dichloromethane sit at the bottom of the water table in an anaerobic environment. And so it makes it a little bit more recalcitrant than one would expect for a molecule of its structure and its size. In 1996, the Halobacterium formicoaceticum was discovered, and it was the first bacterium that was known to anaerobically degrade DCM. That bacterium was noted to do that through the pathway shown here on the right, the Wood-Lundahl pathway, which was just a way for carbon-1 C1 compound metabolism to proceed to either small organic compounds or CO2 through a folate-dependent pathway. The Wood-Lundahl pathway is used in a number of organisms, and part of our interest in this was to actually understand how the chlorinated compounds became loaded onto the folates to enable this pathway to move forward. There were three hypotheses from that. One was that you would go through a cobamide-dependent dechlorination, which would just lead C1 compounds that weren't chlorinated that would feed into the Wood-Lundahl pathway. Another one was that the chlorinated compounds would be added directly to the THF with the chlorine still intact. And the third hypothesis was just that there was an unknown enzyme that supported dechlorination and loaded the tetrahydrofolate. So we started a study with the Loeffler lab looking at a variety of organisms, including this consortium, Candidatus dichloromethamonas elquinensis. I apologize, I'm a chemist. I'm going to mess up every one of these names. I practiced them all night. I'm sure they're still terrible. So you're just going to have to bear with me. So this organism is actually only able to grow in a consortium called RM and was recently discovered. Now, I will tell you that we've done a lot of mass spec, a lot of metabolomics, and a lot of things looking to try to find these folate compounds, either chlorinated or unchlorinated, because we were very excited about these hypotheses. None of those experiments led to the magic structure that told us exactly how things enter into the Wood-Lundahl pathway. But a number of those experiments actually revealed interesting differences in the way that the Halobacterium and the consortium use their Wood-Lundahl pathway. So one of the things that we did was to feed carbon-13 to either the isolated Dichlorobacterium or the consortium in an NMR tube that had been sealed, and actually monitored the metabolism of the living organisms in the NMR, right? So one of the interesting things about dichloromethane when it's carbon-13 labeled, not only does it allow you to see this relatively hard to detect isotope very well by carbon-13 NMR, so you can determine which carbons are labeled. It also has a signal in the proton NMR where the protons are split by the carbon-13 that is next to them. So for instance, what we see in the halobacterium is the small organic acids both have a singlet shown here in the middle from the small organic compounds that are unlabeled, plus DCM is being incorporated, and we get this doublet on the outside, or the split peak, that has to come from a C13 labeled compound. The fact that the abundance of the C13 in the C13 in the molecule is so high shows that there is incorporation. That's also shown in the carbon NMR here, where glycine, acetate, and methanol are highly enriched in C13 label. However, if you perform the same consortium or the same experiment with the consortium, what you see that all the small organic molecules that are detected, methane, acetate, and methanol, are all singlets as expected and have no detectable C13 incorporation. If you look into the carbon-13 NMR and you squint really hard, you may see a little bit of C13 in methanol, but instead what you see is that the majority of the carbon-13 that came from the dichloromethane is instead in carbon dioxide and carbonate. This indicates that this organism is not using dichloromethane to build small organic molecules for other biosynthetic processes, and instead it is mineralizing the compound. That's actually intriguing because until this study, people assumed that mineralization was a process that was energetically unfavorable compared to fermentation. However, if you go in and do the free energy calculations, you can see that at low hydrogen concentrations or low partial pressures of hydrogen, mineralization actually becomes more favorable than fermentation. So what we think that is happening is that in D-formicoaceticum, we're using both ends of the Wood-Lundahl pathway to make both organic acids and CO2, which ends up making acetate and formate, as one would expect from the Wood-Lundahl pathway, to where in this Candidatus species, what we are seeing instead is a co-metabolism where they are mineralizing dichloromethane to CO2. The hydrogen production is being used by some methanogens, and the CO2 is being used by acetobacterium to make the acetate that you see in the culture. This is intriguing because it just, to us, indicates that there are very different ways that pathways are used depending on the environment and who the partners of organisms are. So from there, once we realized that we could start to get relatively good information on consortia of bacteria, we decided to move and look at a different consortium that was capable of the reductive dechlorination of perchloroethene to trichloroethene to cis-dichloroethene to vinyl chloride and then to ethane. These PCEs are major contaminants, and most of our Superfund sites in the US actually have a PCE contamination component. I'm not going to put how much is produced each year because it's been curtailed significantly because of the problems of it, but almost every major industrial site from the 50s, 60s, and probably the 70s, and every military base that was operating in those times has major plumes of PCE underneath of them, which is an issue for a number of reasons. Now, we do know that there are organisms that can do reductive chlorination processes that were discovered in the 80s in these chlorinated ethene plumes underneath these sites. And they can do that either aerobically or anaerobically through respiration or a number of other processes. What also has ended up being very useful after the discovery of these organisms is that you can inject them in the ground and use them to actually perform dechlorination and move to ethene. However, one of the problems is that as the PCE moves in the groundwater and is converted to TCE and to ethene, you actually end up having a step where you become more toxic when you made vinyl chloride than you would have been had you just left the PCE in the environment. And unfortunately, there tends to be a stall at this stage in the dechlorination. So if your bioremediation efforts aren't well monitored and aren't proceeding, you actually end up toxifying the environment instead of detoxifying it. And one of the reasons that happens is because the bacteria that perform the first few steps of the dechlorination are not the same as the Dihalococoides species that perform the final few steps of the dechlorination. Further, the Dihalococoides requires metabolic products or byproducts from the earlier species to really thrive. And therefore, if it does not get what it needs from the consortium to start growing, you have this stall of these more toxic intermediates. And so we decided to ask the question, could we use metabolomics as a monitoring tool in the environment to differentiate the different stages of dechlorination? And so we went and we looked at these consortium SDC9 under a number of conditions, either fed cis dichloroethane or vinyl chloride. And just to show you the standard measurements that one would make, you have to initiate growth by adding lactate. Lactate quickly goes away. Once lactate goes away, you can see that, or the lactate leads to a total growth in bacteria. Once the lactate is depleted, overall bacterial numbers drop if you're utilizing either cis-DCE or vinyl chloride as your organic substrate. However, you can see that the Dihalococoides species, DHCs here, actually start to really thrive once lactate is consumed and the environment has been primed for them by the other bacteria. So once we determined sort of how the bacteria were going to grow, we also monitored the concentrations of the chlorinated ethenes and ethene over the course of the experiment. As you can see, if you add cis-DCE, it quickly degrades. Vinyl chloride is produced and it begins to degrade as well. And over time, ethene is produced. If you add just vinyl chloride, it degrades relatively quickly with the production of ethene. And so when we go in and look at the metabolomics of this, we decided to look at several different time points corresponding to the ratio of vinyl chlorides that you see here before, or see here at the bottom, where we wanted to sample when metabolism was primarily utilizing cis-DCE, or metabolism was primarily utilizing vinyl chloride, and where dechlorination was over. And TX is 3 days after there was no detectable chlorinated compounds, and T5 is 1 month after. Just to give you an idea of the number of things that we detect, if we look at all features from the water-soluble metabolomics experiments, we detected between 9,000 and 11,000 things in each of these cultures. Once we de-isotoped and de-adducted, which is just a way to remove things that are the same, about 75% of those compounds seem to be unique, potentially biogenic molecules, and we were able to identify several hundred of those. Again, this just highlights sort of the knowledge gap, and in my opinion, the exciting part of metabolomics is that even though we're detecting more than enough things to make biological conclusions, we're still learning a lot of new things about metabolism as well. So from there, we use partial least squares discriminant analysis, which is a close cousin to PCA, to try to see if the metabolome was able to cluster these different regimes of dechlorination. And what we found is that T initial, which was before dechlorination started, and T5, which was well after dechlorination ended, clearly were different from everything else and looked similar. Regimes where we were primarily utilizing cis-DCE were also well separated. And then the points where we had shifted to primarily using vinyl chloride, or we're probably using residual vinyl chloride that couldn't be detected, also clustered very well together. And so from this, we believe that, at least in the laboratory, we can go in and determine how these cultures are functioning and potentially predict stall just based on metabolomics and their metabolic fingerprint. The same holds true for vinyl chloride, but in the sake of time, I won't go through this in detail as well. So where we are now on this project is actually going out into the field and collecting samples from Superfund sites and industrial sites all over the nation to see if we can find the same signatures in the environment that we have found in the laboratory, and hopefully to reduce the number of things that we need to detect to a quick test that people interested in bioremediation can use to determine whether or not their system is stalled. We also hope that we can learn more about the metabolism of these species and maybe even understand why stall occurs. But the complexity of this is going to take a while for us to tease out those details. Now, the last talk, we're going to switch from discussing chlorinated compounds to discussing fluorinated compounds. If you at all follow the environmental sciences world, you'll know that the emerging contaminant that many people are most worried about now are these perfluorinated alkyl sulfonates and perfluorinated alkyl carboxylic acid compounds, the PFOSs and the PFOAs. And the reason that people are worried about these is because the carbon-fluorine bond is much stronger than any other carbon-halogen bonds, and that it's very hard to break. Many people call them the forever chemicals. They're forever contaminants, and they're what makes Teflon. They're also used to put out fires because they don't burn and don't degrade that way. And they are, the more places we look, the more places we're finding them. Now, there's a debate on whether or not you could even perform bioremediation of these compounds because of the strength of the carbon-fluorine bond versus the strength of the carbon-chlorine bond. And the Loeffler group, along with us and a few other groups here in Tennessee, as well as groups all over the world, are trying to find a way to deal with these compounds. So I'm going to quickly go through and just talk about one of our efforts using Pseudomonas species strain 273, which was shown to defluorinate 1-dichlorodecane and 1,10-dichlorodecane several decades ago. Now, this isn't a perchlorinated compound, so I don't want to make anyone think that we're going to utilize the same mechanisms here, but I just want to point out that there are other fluorinated compounds that are also a problem. So the Loeffler lab utilized this compound and showed that it could degrade 1-fluorodecane and 1,10-difluorodecane, as well as the chlorinated compounds. Just for full disclosure, this probably isn't through direct cleavage of the carbon-fluorine bond, but that we can handle that in the question if anyone is interested. And so once they figured out this happened, they asked us to do metabolomics and lipidomics experiments to see if we could understand what was going on. I'm not going to talk about the lipidomics experiments today, but I do want to talk about the lipidomics experiments because we found something that was actually rather surprising at the time. If you look at the cultures grown on decanoate, decane, 1-fluorodecane, and difluorodecane, all of them grew. But when you look at the lipidome, what was interesting is that the lipids themselves became fluorinated the more fluorinated the compound was. Now, this was an intriguing result because as far as I know, this was the first time that conversion of these compounds had been shown, fluorinated compounds to fatty acids had been shown, and they were also making it into the lipidome in a large amount. And this just shows how contaminants impact things in sort of ways that we may not understand, because the lipidome in this organism is over 80% fluorinated at this point. That's intriguing because it probably changes a lot of membrane properties that we don't quite understand, and there are probably consequences of this that are much different than one would expect. Just to show you some data, as you can see, we can mass spectrometrically detect fluorinated compounds when fluorinated substrates are added, and we can determine that they're fluorinated by shifts in mass. And just to really nail down well where the fluorination occurs, we also did fatty acid methyl ester analysis where we stripped the acyl tails from the head groups, and you can see that the fluorines are in the acyl tails on these and not in the head groups. This publication should be coming out shortly that reports not only the fact that this difluorodecane can be degraded by bacteria, but also shows sort of the unexpected fate of these byproducts in this organism and in the environment. So with that, I would like to thank everyone for their attention and also thank the people who either performed the work or did the work. These are my current group members and then some select former graduate students who have moved on to other things. People highlighted in bold: Amanda May, Alex Fish, Abigail Tester, and Alan Borden directly had their hands on generating a lot of these data. Dr. Hector Castro is the manager of the core facility here, who is instrumental in keeping technology running. And Eric, Caleb, and Josh all developed some sort of technology that was utilized in at least one of these studies. The collaborators from the Loeffler lab are Frank Loeffler, Gao Chen, Jun Yan, and Yangqi Zi. And then we also have some wonderful collaborators at Microbial Insights, a local company, Dora Taggart and Sam Rosalina, who have helped with this. And then Mandy Michelson from the Army Corps of Engineers has also been involved with these studies as well. And with that, I would like to thank everyone again for their attention and would be glad to answer questions.
This has been awesome. Thank you so much for sharing this work.
Thank you.
We'll invite everyone to type in their questions, and I see that we have a couple. But while I'm giving everyone a minute to add additional questions, I'd love to just start with asking you a little bit about, I have never personally been involved in bioremediation projects. And so I'm curious if you could walk us through some of the approaches at a practical level that are used for these projects in terms of, are we adding some of the microbial consortia that you've discussed directly to sites? Is it often supplementing microbial communities that are already there with molecules or enzymes? How does that work?
It's, I mean, both are doable, but usually what happens is an injection of the consortium SDC9 into the site, or stimulation of the bacteria if they're known to be there. But yes, you can actually buy 20-gallon or 20-liter buckets of this stuff, and they'll give it to you if you're an academic, and then they just take it out and inject it into the ground.
Okay,
right?
And so it's being sort of injected or applied at a site. And does that also apply when it's when you're trying to get to that DCM layer as well beneath the water table that's sort of anaerobic? Are we trying to inject directly into that layer?
Yeah, these tend to inject, and really the biggest issue isn't injecting the bacteria or the consortium of bacteria. The biggest issue is keeping them healthy and happy and injecting the right mix of cobalt species, cobamides, other electron acceptors. There's a lactate. There's a whole lot of amendments that people try to add to them to keep them going and functioning and healthy. And that's really the knowledge gap for bioremediation, in my opinion, is the amendment and monitoring rather than the application.
Sure. Okay. Let's see. We've got a question from Rachel here. Who says, amazing work. The NMR spectra of the labeled DCM metabolism is really beautiful. It seems remarkably clean with few peaks. Is that a direct reading of the tube while the bacteria grow? Correct.
Yes, it is. Yeah, that is bacteria in a tube. It's sealed. Oxygen was pumped out, and they are growing in the NMR tube. And if you go look up the paper, we actually have a time course where we followed the culture every couple hours for several weeks, and you can actually see the shifts in metabolism as it moves forward.
Awesome. And I don't remember, was that one of the consortia, or was that a single organism?
It's both a single organism and a consortium.
Okay.
There's comparative.
Okay, great. Chelsea, I realized I should just also invite you, if you want to jump in at any point with questions, feel free to do so. You're welcome to just pop in here. Let's see what else we've got. All right. I know there must be some more questions from the audience, so I'm gonna just prod you to feel free to type them in. We want to hear some more from you and continue the discussion here. Meanwhile,
Can you hear me?
yeah, go for it.
Okay. Shawn, I'm curious how durable or how long would the fluorines on those fatty acids remain if you remove that contamination? Is that a stable thing, or is that something they'll try to get rid of quickly?
I don't know. We haven't looked into that, right? Yeah, it's a good question.
And do you think though that it's changing the shape of the membranes?
I don't know that it's necessarily that in and of itself is changing the shape of the membrane. There's a little bit more data that we didn't talk about because of the length here. But it also, the fluorinated alkanes seem to lead to a higher level of unsaturation in the membrane as well, which is definitely going to change the shape. I think the fluorines are going to change the electrostatics of the interactions at the tails, right, when the two fluorines are close together. But the level of unsaturation is also going to cause differences. And metabolically, what we haven't really pinned down yet is whether or not we're oxidizing one end of the chain, which removes or leads to spontaneous release of the fluorine. And then is that fatty acid on some sort of acyl carrier, cofactor, or protein that's elongated without further degradation? Or are we degrading the alkane all the way down to fluoroacetate, and fluoroacetate's being loaded into lipid biosynthesis pathways and re-elongated? And we don't know that. And there are slightly different potential outcomes depending on which way you think we're headed.
Yeah, I was curious also about whether whether you've looked at the physiology of the microbes after their lipidome has changed that much, and whether you can either visually see or detect through different assays any changes there.
We've run the experiments, but we haven't analyzed those data. We're excited about the lipid work, and we also went down a little bit of a rabbit hole looking at how the PFOSs and PFOAs are either incorporated into or changed to lipids as well. So, yeah.
Awesome. We have a question from Minwoo. Great talk, Shawn. I noticed the process of TCE bioremediation takes months in the laboratory setting. I'm curious if the rate of bioremediation is comparable, slower, or faster in the environment.
It is consortium dependent. And sometimes we get SDC9 from the supplier and remediation is over in the course of weeks. And sometimes the consortium takes months. And we don't quite understand what the differences are there. But in the environment, it is fast enough to fully contain plumes of fluorinated compounds at sites, which is the important part, right? So.
Awesome.
Yeah, I know that's not a super quantitative answer, and I apologize, but it is a highly variable process.
Sure. So I'm curious, I know that you had mentioned that there are some restrictions on what you can share, but there was definitely a little bit of a teaser in terms of some of the forensics work. And so I'm curious if you could just verbally touch on a little bit of your work there.
Sure. So the forensics work actually looks structurally a lot like our work with SDC9. And so we have the Forensics Anthropology Center here that has a capability, and I'm trying not to call it its colloquial term, which they don't like, has a capability to put human cadavers out into the field and understand how they decompose over time. Mm-hmm. And what they have found is that they were bringing in bodies of the same relative weight, same relative age, same relative conditioning. Some of them would be almost fully skeletalized in a couple of weeks to a month, and others would have most of the flesh left on them for months, right? Much longer periods of time. And the only really variable that they could go in and figure out that was different was the pre-mortem toxicological loading. And so we've done a study where we're looking at the interactions of drugs and sort of what drugs do to the decomposers. primary decomposition happens through insect consumption, scavengers. In Tennessee, that's primarily raccoons. And then a lot of it is microbial processes. And the data that I couldn't show was a picture of a diabetic patient that after 17 days is almost fully decomposed, and a picture of a cancer patient that after 17 days, other than some discoloration, looks like they've just been laid on the ground, right? And so we've got a huge study following maggot metabolism of the decomposition fluid that comes out of the cadavers, and also the soil communities underneath of the cadaver, to see how changes happen. And it looks like there's been a couple really interesting things. First off, humans, when we die, are a major source of contamination in the environment. We put off a lot of drugs into the environment that alter microbial metabolism, alter scavenging, and alter larval development in real ways. And we're trying to tease out exactly how that happens based on the types of drugs, so that, if forensics investigations are needed, we can in the future predict how toxicology determines postmortem time of death, postmortem interval before decomposition.
That's awesome.
Yeah, it's an interesting project. I have a weak stomach, so.
So challenging too.
Yeah, well, no, it's not challenging for me because I sit here and they bring samples back. But students really like to go out there and sort of see the processes. And the Forensic Anthropology Center has been a huge resource.
Mm-hmm. Yeah, absolutely. And how much overlap are you seeing between these projects, in the sense that do there seem to be similar organisms or consortia or processes that are mediating the processes that are coming during that human biological breakdown, as are mediating some of the processes of breaking down these chemicals at other sites?
We're only looking in the surface layer of the soils, and so most of them are aerobic organisms versus anaerobic organisms in the bioremediation. So.
Awesome. Well, I will put out one last call here for any remaining questions before we wrap up. Give it 1 or 2 more minutes for anybody in the YouTube audience. But meanwhile, thank you. This has been really informative and awesome in a subject area that's largely new for me. And so really interesting to hear all of your work.
Yeah, no, thank you for the invitation. It's always fun to talk to people from different disciplines and move out of the chemistry world for a little bit.
Absolutely. Let's see. All right. Well, I think we'll sign it off here. I'm going to take it off of YouTube Live now. And so we'll disconnect there and then say goodbye offline or behind the scenes, I guess.
Yep.
So
we'd like to thank everyone again for coming, and thanks for listening to our science. So.